It's 12:30, so let's get started. The first thing I want to do is go over quiz 5. So let me share my screen with you. Here's quiz 5. Let me know if you can't read it. So I'll go over this quickly. Just want to make sure you know what the correct answers were and where did the questions come from. So the first question was, mutations can be caused by what? And you choose from this list. And I have red X's here for the correct answers. CRISPR is the ultimate in causing mutations, but they can also be caused by UV radiation, chemicals, and errors in replication. And that question came directly from this slide in the lecture on mutations. Then the next question was, decipher the gene location designated by this code. And the interpretation would be that there's a gene on chromosome 16, long arm, region 23, band 4, subband 1. And that came from the section on CFTR, where we talked about the code for that particular gene. Then the next one was multiple choice, indicate the correct sequence of events. And that came from this figure where we have the gene, CFTR gene encoded in the nucleus. The protein ends up in the membrane. So the correct sequence of events was, first you transcribe the mRNA in the nucleus, then the protein is made and glycosylated in the Golgi, that's here. Then it's transported in vesicles and ends up in the membrane. And finally, the fourth question was about recognizing types of mutations. And this came directly from this slide in the mutation set. I showed you 4 pictures of chromosomes and asked you what type of mutation is represented by each. So number A is a translocation. We have these 2 chromosomes exchange tips, and that's called a translocation. The next one is an inversion, where an inversion is a piece of DNA is flipped over. See that here. And the next one is a duplication. There's a section of DNA on this chromosome, which is copied and repeated. And finally, the last one is a deletion, where you have this green section of the chromosome that's just missing in the mutant.
I have a quick question.
Yeah.
What's an example of insertion? Because I put that as an answer for the first, or like the third one, and I got it wrong. But I retook it and got it right again.
Let's see. Faraj, okay. What is an insertion?
Yeah, just like an example of one too.
An insertion could be as little as 1 base pair. And if it were 1 base pair, it would be a translocation. It would cause a frame shift. Or it could be anything more than 1 base pair. It could be 2 or 3. It could be 1,000. It could be 1,000,000. Insertions come in all sizes. And they have a dramatic effect. So for example, a 1 base pair insertion causes a frame shift And that changes all of the amino acids from that point on. So it makes sense?
Yeah, that makes a little bit more sense. Thanks.
Yeah, here in the description, insertion, I've said it's a new piece of DNA introduced. And that piece of DNA could be one base pair or anything larger than one base pair. All right, any other questions about quiz number 5?
Yeah, you said the on the first question, what a mutation was. Did you say CRISPR was one
Yeah.
of them?
Yeah,
Because I did all the other ones, but that one I didn't, or I thought I marked it, but I guess I didn't, so I got that wrong.
yeah. CRISPR, I hope you all got the point that the way to make mutations, if you want mutations, the way to make them these days is to use CRISPR. Because remember the 2 points about CRISPR? One, it's universal. That means it'll work in any organism. And it's specific. You only mutate the target that you've chosen. Anything else on quiz 5? If not, I'll take it away. And next, I want to share my screen again with you. And go back to coronavirus. Remember, I had a problem. I couldn't find the correct slide set when I showed this to you the first time. So the slide sets are largely overlapping, and I'm not going to go over the entire thing again, but I want to make a few points that the new slide set makes and the old one didn't. So you remember, how does the virus enter a cell? You remember it has the spike protein, and the spike protein binds to this receptor called ACE2. And then it enters the cell by a mechanism called endocytosis. And that's illustrated a little better in this slide, where the binding of spike to the receptor is a stimulus that causes the cell plasma membrane to invaginate and enclose around the virus particle so that it ends up inside the cell in a vesicle. And then from this vesicle, it's released. And if it's an mRNA vaccine that we're talking about, then the mRNA is released and is transcribed and translated. So that is one point I wanted to make. Then here is another one. Why is the virus so highly infectious? There are thousands of SARS type viruses. And why is this one causing a worldwide pandemic? Well, one reason is that the spike protein binds to the receptor ACE2 very tightly, about 10 times greater than with other SARS type viruses. So this means that only a few particles are needed to start an infection. I'll talk a little bit about the Delta variant. That's the one that's causing so many problems right now worldwide. It's even more infectious than any other strain. And the other thing I want to emphasize here is that this is an unusual virus in that it has a 2-week latent period. That is, when a person first contracts the virus, it begins to multiply in their respiratory system, but there are no symptoms. And there are no symptoms for about 2 weeks. So during that 2-week symptomless period, this infected individual doesn't know that they're infected. So they're going around intermingling with other people, spreading the virus without knowing they're infected, without knowing they're causing other people to be infected. So during this period, they're known as symptomless carriers. And this is why testing is so important among people who are not vaccinated, because they could be carrying the virus and spreading it around without having any symptoms. But if they're tested and shown to be infected, then they know and they can quarantine.
Professor, can people who are vaccinated still have COVID and spread it without knowing, even though they are vaccinated?
Yes. Let's see. Yeah, people can have that are vaccinated can have virus that does replicate, but at much lower levels usually. So that's one argument for why people who are vaccinated, when they're in areas of high concentration of other people, when they're around groups, they should still wear a mask, just on the outside chance that they are carrying virus and don't know it. Anything else on the infectiousness of this virus in general? All right. Now, the thing that we worry about most is new strains. They're called variants. Well, how do new strains arise? There are two main ways. One is mutation, and the other is recombination. They're illustrated in these two pictures. On the right here, as the virus is replicating, there are copying errors, always. And these copying errors lead to new amino acid sequences. And so here you have a wild type, and then here the little red bar indicates that a mutation has occurred. So that's one way. The other way is recombination. So suppose that two different variants of the same virus infect the same cell. And while they're inside that cell, they recombine, they exchange parts of their chromosomes. Now you have a new virus, which is a hybrid. It contains part RNA from the blue strain and part RNA from the red strain. And so that's a mutation, and that could have a whole different set of infection properties.
Professor, this is Steve Stinson.
Okay.
And my question is, especially with the recombining, can a virus or mutation, virus such as the COVID be made to recombine and therefore mutate quicker? Can it be designed to do that, especially since we got CRISPR?
It couldn't be designed. You're thinking of in a lab? You could certainly recombine the genomes in a lab under contained conditions, but what this is referring to is natural infections. So if you have two different viral strains infecting the same cell, there is the possibility they could recombine their genomes. And if they do, that's a mutation.
Professor,
Uh-huh.
this is Alex Allo speaking. My question is like, is the chance of getting, say, like the Delta variant, like pretty low compared to like, say, the regular COVID?
It's actually the opposite. The chance of
Higher?
getting Delta is higher than other strains. And I'll talk a little bit about that in the next slide. But let me just finish this slide first. At the bottom here, remember copying errors and recombination. That's how we get new strains of coronaviruses. And these processes occur continuously. So that means that the more unvaccinated people there are, the more the virus replicates, the more mutations occur, the more genetic variation, and the greater the chance that a mutant will be more virulent. So really the only way to stop this pandemic is to get enough people vaccinated that we slow down the replication, the worldwide replication of the virus.
From what I was told, I think I heard that San Diego was 70% vaccinated.
I think that's about right. Yeah, there are some sectors of the population that are higher. There are other sectors that are lower, but I think overall, even nationwide, it's about 70%. And for what's called herd immunity, the experts usually recommend 80% or higher.
I see.
Okay, I showed you this slide before. It just shows the viral RNA genome. And mutations can occur anywhere in it. This red section is what codes for the spike protein, and that's where mutations would be the most dangerous. And then if you look worldwide, see that there are thousands of mutations. So in each of these lines, that means that the genome of the virus from an individual person was sequenced, and its sequence was compared with all the other coronaviruses that have been sequenced. So the Delta variant, it's a mutant in this group, and this shows that they come from all over the world. And now let's talk a little bit more about the Delta variant, because that's the one that has really emerged and taken over the population of coronavirus worldwide. So how did it originate, and why does it have increased virulence? Well, it has mutations in the gene encoding spike. Spike, you remember, is the part of the virus that binds to the human cell and gets inside. And these mutations in the spike protein are the result of 3 amino acid substitutions. And they're indicated here: T478K, P681R, and L452R. These codes are shorthand for telling you the position in the amino acid chain and what was substituted for what. So 478 means that amino acid position 478, there's normally a threonine. And in the Delta variant, it's replaced by lysine. And just to remind you, there are single letter and multiple letter codes for amino acids. So T to K, T is threonine, change to K, which is lysine. And another mutation, P to R. So if we look, P is proline, that's the normal one. It's changed to R, which is, where is R? R is arginine. So proline is replaced by arginine, and leucine is replaced also by arginine. So these 3 amino acid substitutions are what's different about the Delta variant. And these substitutions are known to increase virus transmissibility. That is, it's spread from person to person more readily than any other variant. This variant is the most transmissible of any respiratory virus known yet. So, and these 3 amino acid substitutions cause it to bind more tightly to the ACE2 receptor. So the Delta is about 50% more contagious than the original strain, which is called Alpha. So that means that only half the dose, you only need to inhale half as many particles to start an infection. So it's more transmissible, but the symptoms caused by Delta are not necessarily more severe. It seems to cause about the same symptoms as any other strain. It's just more transmissible. And as you probably know, the symptoms are highly variable from person to person. Some people have only mild symptoms, some people have no symptoms, some people die.
I have a question. This is Sean Koch.
Okay.
So yeah, something I was wondering is there, have we found out any reason why it has such a wide variety of symptoms? I mean, I've read that some people just experience a mild flu, whereas other people end up getting neurological disorders from it. Other people have talked about this long COVID, where they feel entirely drained for weeks, sometimes even, months after having contracted it. Is there some reason why it has such a variety of symptoms?
Yeah, there's probably a lot of reasons. And the reasons are basically the differences between people. So for an extreme comparison, take someone who has a normal, healthy immune system and contrast that someone who's had an organ transplant and they have a suppressed immune system. The latter is likely to get much more severe symptoms than the former. So it just depends entirely on the physiological condition of the patient as to how severe the symptoms are going to be. The number of people, the percent of people that actually die from COVID is relatively low, but the number of people who get symptoms of one kind or another is fairly high. There are not many, percentage-wise, not many symptomless carriers. Okay, anything?
I got a quick question. This is Anthony. It says that these amino acid substitutions are known to increase virus transmissibility. Why do they increase transmissibility? Is it just the T to the lysine? Does lysine cause it, or what exactly does it do?
Okay, so what we have here is spike binding to ACE2. They're both proteins.
Mm-hmm.
So you have amino acids binding to amino acids, and it just turns out that these 3 substitutions bind more tightly to ACE2 than the original strain.
I see.
So that means that the dose that it takes to cause an infection is much lower, about half. You need about half as many virus particles. You think if you're out in the open around other people and they're coughing or sneezing or making aerosols that have the virus and you breathe it in, you only need half as much of a dose to get infected if it's the Delta variant.
Okay, so it's not necessarily that it's lysine, it's just that it's within the Delta variant, these specific ones cause that.
Yeah, these specific ones change the binding properties.
Got
So
you, okay.
it binds more tightly.
Okay, because I had a question regarding the COVID. Since the COVID-19 came out in 2019, and now they're having different other viruses that come out, and they're saying they're connected in some way to the COVID, which is like the Delta, and then there's another one they say they contracted or whatever found. Okay, but before, when we had the flu, every year there was like a different flu that was tested that was out, and they made new vaccines that you had to take flu shots that would cover different ones or whichever ones they said was going to be out for that year. Is this the same scenario with the COVID? Is it going to be like that continuously like that?
With COVID, we just don't know. We've had flu viruses around for long enough that we know something about their infectivity, and they're monitored. The flu viruses are sequenced continuously. So we know the variants that are being produced by flu virus. And so the new vaccine that comes out every year, always in the fall, is based on the sequences of the flu viruses that are present at that point in time. Now, in the case of coronavirus, it's not seasonal. It spreads equally in all seasons. So we can't just say, okay, get your coronavirus vaccine in the fall or in some other season, because it doesn't work that way. It doesn't respect seasons, whereas flu viruses are almost always worse in the winter months than they are in the summer.
Oh, okay. Okay. Thank you.
Okay.
Oh, Yolanda, by the way.
Okay, the rest of this slide set is about the vaccines. Are there any questions about vaccines? I won't go through it all again unless somebody has questions about it. Okay, and again, you have the recordings of both last time and this time. So if you want to go back and review. So let me stop sharing the coronavirus. And next, I want to introduce a new topic. Membranes. Membranes and transport. What is a membrane? It's a selective barrier that allows some things to go through and stops others. Transport means the movement of something from one point to another. The plasma membrane is a very special one. It's a phospholipid bilayer that contains proteins, and it forms a barrier between a cell's contents and its exterior. This picture shows two cells, cell 1 and cell 2. Each cell has a plasma membrane, which you can see in this electron micrograph. And you can actually see the lipid bilayer here. You see the little space between the two lines. It looks like a railroad track. This is the lipid bilayer of cell 2, and this is for cell 1. And there's a space between the cells, between the two lipid bilayers. So why are we making a big deal about membranes? Well, life depends on cells. And cells depend on membranes. Cells could not exist without them because they partition life from the rest of the world. In other words, what's going on inside a cell is alive, and that has to be kept separate from everything going on outside the cell. So membrane functions, they're semi-permeable. They control exchange of materials. We'll talk about that. Communication, cell-to-cell recognition. Later, when we talk about energy, we'll see that energy transformations occur within the membrane and cell adhesion. Now, what are the molecular components that make up the plasma membrane? Well, there are 4 major classes of biological macromolecules. Remember what they are? The plasma membrane contains 3 of them, but 1 is missing. Can you think? Here are the 3: lipids, proteins, and carbohydrates. These are all components of the plasma membrane. What macromolecule is not there? Anybody want to guess?
Fat.
Remember when we talked about most of the mass of a cell is made of these 4 different components, and 3 of them are lipids, proteins, and carbohydrates. What's the one that's missing? What's the genetic material? Nucleic acids. Nucleic acids are involved in replication of life, but they're not involved in membranes. So that we'll talk more about the lipids, more about the proteins and the carbohydrates later on in this slide set. I just make a note here that there's a extracellular matrix called the glycocalyx that is composed of proteins and carbohydrates. And this gives you a better picture of what the glycocalyx actually is. It's a thick gel-like covering of the plasma membrane. It's composed of sugar and protein polymers bound together. So you see on the right here a picture. Well, at the bottom left, you see a cross-section of a bacterial cell. And you see its surface looks fuzzy. And if you take a closer look at that fuzzy section, you see it looks like little hairs. And these are composed of polymers of sugars and amino acids. So proteins and complex sugars get all tangled up together and form this layer called the extracellular matrix or the glycocalyx. And this covering of the cells is involved in a lot of different cell functions: stickiness to a substrate or stickiness to each other, keeping cells hydrated. For example, the moistening of the corona. If you ever caught a fish and tried to pick it up, it might have slipped out of your hands. It was slippery because of its extracellular matrix on the scales. And this just gives more detail about the extracellular matrix. Illustrates again what the strands look like. And now let's go on to the phospholipid bilayer membrane. This is the membrane that's involved in life. So it's constructed from phospholipids. There are 2 layers. The outside of the layer is hydrophilic, it's water-loving. The inside is hydrophobic, water-hating. So first, let's look at the colored picture here, the blue one, where you see the bilayer, and you see on the left, the phospholipid molecule, which is a long carbon chain, a hydrocarbon chain, which is non-polar. And then there's a polar head, which is composed of a phosphate group, and then another hydrophilic group. And this can be variable. So this membrane is essential for life because it separates the watery cytosol, that's where all the living processes go on, from the watery exterior. Most cells find themselves in an aqueous environment. They're always aqueous on the inside. They're usually aqueous on the outside. So the membrane has a hydrophilic layer on the outside, 2 hydrophobic layers in the center, and then another hydrophilic layer on the inside. And just to remind you about what a phospholipid looks like, it has these hydrophilic heads and then these long hydrophobic tails that can be either saturated, in which case they're stiff and rigid, or unsaturated, which means they have flexibility, they can move around. And here up in the upper right are the structures of a saturated and an unsaturated lipid. And I've got a note here at the bottom of this picture that phospholipids are amphipathic molecules. And we find amphipathic molecules in a number of places in biology. And I'd just like to remind you, when we talked about the pancreas and the gallbladder, again, we ran into amphipathic molecules. If you recall, the gallbladder produces bile, which contains these bile acids, which are composed of a steroid, which is very hydrophobic, and a hydrophilic tip on the other end of the molecule. So this, because one end is hydrophobic, the other end is hydrophilic, that makes it an amphipathic molecule. And you remember why this is important. It allows us to eat fats. If it was not for these amphipathic acids, we couldn't digest fats. But because we have them, fats can be a major part of our diet, and we have no problem with them. Another place we run into amphipathic molecules is with soaps. Soaps are amphipathic molecules in action. So a soap is a surfactant. It breaks the surface tension. And a soap is always composed of a hydrophilic head and a hydrophobic tail. And if, for example, you have oil in water and you pour in soap, what is formed are called micelles, where these hydrophobic tails go into the oil and the hydrophilic heads remain in the water. So you might ask, what's the difference between a soap and a detergent? Well, there's not much difference. They're both amphipathic molecules. They both clean in the same way. They both have long hydrophobic tails, but they differ in their hydrophilic heads. And one of the reasons for using a detergent rather than a soap is that they don't precipitate cations like calcium as much as soaps do. So in hard water, detergents are more effective. So soaps are interesting because they're so simple. What is a soap? Well, historically, people mixed ashes with cooking fat, lard, and got soap. So the recipe is really simple. Go to your fireplace to get some ashes, boil them in water, add some fat or lard, and pour it into molds, and you get soap. And here you see pioneer women with big pots containing these substances, making soap, and it can be hardened and cut into bars, and this is primitive soap. On the lower left, you can see that this process was done on a commercial level, these large vats. So let's take a closer look at how a soap molecule is actually
Now,
made.
this is what they did then, right? Not now.
Yeah, well, soaps are still made in the same way, but much
With
much
ashes
more
and lard?
sophisticated modern process. But
Okay.
it still comes down to hooking up a hydrophobic tail to a hydrophilic head. And so
Mhm.
how has this been done? If you look at in this blue box, a fat, you recall, fats are triglycerides. So this is the structure of a triglyceride, and that's hydrophobic. Now, ashes are essentially sodium hydroxide, NaOH. So if you have one triglyceride molecule and 3 sodium hydroxide molecules that came from the ashes, they combine to make a soap. 3 molecules of soap, a long hydrophobic tail and a hydrophilic head. If we look at the structure of an example of a soap, sodium laurate, you can see that in the space-filling model, it has this hydrophilic end, one end, and a hydrophobic tail at the other end. So this is the chemistry of how a soap is made. What happens when you wash your hands? You got dirt on your hands, you add some soap, and what happens is that the nonpolar ends of the soap molecules attach to the dirt. The dirt tends to be nonpolar. Then you add water, and the hydrophilic ends go into the water, and that lifts the dirt off your skin.
In other words, that's what makes the bubbles when you wash your hands,
No,
the lather,
the
when you wash your
bubbles,
hands, the last the yeah,
the bubbles are just formed
that
by the
lather,
soap
the white
molecules
lather.
accumulating. Yeah, no, that is not part of the cleaning action. You could do the cleaning without the bubbles.
Really? Oh, okay. So when you wash
But you
your
always
hands,
get bubbles because that's the nature of a soap. In water, it will form spheres, which I think, yeah, micelles. So you get these essentially bubbles. They can be of any different size. Okay, steroids and glycolipids. Plasma membranes have they have this bilayer phospholipids, but they almost always also have cholesterol. And they also always have proteins, and these proteins can either go all the way through the membrane. So example like CFTR, it's an integral protein, or it can be a peripheral protein, which is embedded on one side of the membrane, but doesn't go all the way through. Now, I want to give you an impression of what a membrane is really like. And the best model of that is called the fluid mosaic. model. A fluid means semi-solid. So the phospholipid layers are fluid. And they are a collage of proteins embedded in this matrix. It's not homogeneous. There are many microdomains in a membrane. So here you can see a picture, a drawing of a membrane, and here you can see an electron micrograph of a similar membrane. So what are the details of this fluid mosaic membrane model? The membrane components, which are proteins and glycolipids, form a mobile mosaic of islands floating in a sea of phospholipids. So we have a phospholipid bilayer, and we have these proteins and glycolipids floating around in this bilayer. At room temperature, the plasma membrane has the consistency of vegetable oil. So it's fluid, and that allows components to float around in it. And they're in constant motion. Membranes, if a cell is alive, the membrane is constantly moving. Now, factors affecting fluidity: as fluidity increases, permeability increases. So at higher temperatures, it's easier for things to get through the membrane than at lower temperatures. So the three factors that you need to remember are temperature, fatty acid content, and cholesterol. So temperature, as I just said, as it goes up, fluidity and permeability go up. Fatty acids, unsaturation promotes fluidity. In other words, double bonds in the hydrocarbon tail make the membrane more fluid. And finally, cholesterol is a buffer. So at low temperatures, it increases fluidity, and at high temperatures, it decreases fluidity. And this is important because of this note. If the fluidity is too high, the structure is not supported and the membrane falls apart. If the fluidity is too low, the membrane loses permeability and molecules cannot get across. So that's why cholesterol is important. It buffers between too much fluidity and too little. And these things are illustrated on the right. Membrane fluidity, if the hydrophobic tails are saturated, it's less fluid. If they're unsaturated, or if some of them are unsaturated, it's more fluid. Here's a temperature curve. As the temperature goes up, the fluidity goes from more solid-like to more liquid-like. And cholesterol, as I said, is a buffer against temperature extremes. So to imagine what a membrane, a biological membrane, is really like, you have to use your imagination. You can go on YouTube and find videos of the motion of a membrane. We won't go into that now for the sake of time. But the phospholipid bilayer is this viscous semi-liquid. The proteins and glycolipids are embedded in this, and they sustain continuous movements. So think of a raft on a pond or in a swimming pool. If the phospholipid bilayer is the raft and you're sitting in it, it would be like one of these proteins sitting in a membrane. So I want to emphasize that all these proteins, some of them are peripheral, they go part way through, some go all the way through. some of the sugars and fats on the surface. All of these things are in constant motion. So the membrane is sort of undulating, moving up and down, and the things embedded in it are constantly moving around. And we're going to talk in a minute about what the real purpose of a membrane, aside from separating the outside from the inside, is to allow certain things to pass through. Okay, and that brings us to diffusion and osmosis. What is diffusion? It's the movement of molecules from a higher concentration to a lower concentration. And you've probably all done this, what's illustrated here. You have a little tube of food coloring, you put a drop in a glass of water, and initially it's all concentrated where the drop entered the water. But wait a few minutes and it starts to disperse. Wait long enough and it'll be evenly dispersed throughout the water. That's diffusion. You can see that illustrated here with molecules. The red are the dye, the blue are the water molecules, and eventually they're evenly distributed. So that is diffusion. What is osmosis? It's the diffusion of water through a semipermeable membrane. So the difference between osmosis and diffusion is that with diffusion, there is no membrane. With osmosis, there is a membrane. Now here we have to talk about two terms, solvent and solute. A solvent is a liquid that can dissolve other substances. And in biology, the solvent is almost always water. The solute is the substance that dissolved in the solvent in water. So you can see the demonstration of osmosis in these figures on the right. If you have two beakers connected by a tube, and the tube has a semi-permeable membrane in it, and this membrane is permeable to water, so water can go through it, but salt can't. So the salt dissolves. Salt is the solute, water is the solvent. Salt dissolves in it. Now, osmosis is the movement of water across this membrane. Salt can't move, but water can. And so it will go from an area of lower concentration of salt to a higher concentration of salt. And that causes the level of water in the beaker on the right to go up and the beaker on the left to go down. So this is osmosis illustrated in the most basic way. Tonicity of solutions. There are three different types of tonicity. One is isotonic. That's when the water concentration outside the cell is equal to the water concentration inside the cell. There are hypotonic conditions. So hypotonic with respect to the environment of the cell, and this is important whenever you're talking about hypertonic or hypertonic, it's with respect to the environment, either the environment of the cell or the environment in which the cell finds itself. Well, hypotonic means that the water concentration outside the cell is higher than inside. And so the consequence of that is that the solute concentration outside the cell is lower than inside. So tonicity always refers to solute. And so hypo means that the solute concentration is low. And we're talking about the outside of the cell. And the result of this is that water moves into the cell at a greater rate than it moves out. Now that's hypotonic. Hypo means low. Hypertonic, hyper means high. And again, this is with respect to the environment of the cell. The water concentration outside the cell is lower than inside. The converse is that the solute concentration outside the cells is higher. So this is with respect to the environment of the cell. So solute is higher, hyper, higher than inside the cell. And that means that water will move out of the cell faster than it moves in. I have these terms here to help you keep the tonicities straight. Osmosis only refers to water. Tonicity only refers to solute. So substance, whether it's the solvent or the solute, either one, will always move from a high concentration to a low concentration. So keep those things in mind, especially, I will assign a worksheet, or maybe I've already assigned it, which will have some problems in it. And to solve those problems, you'll have to keep this guide in mind, that osmosis refers to water, tonicity, hypertonic or hypotonic refers to solute. Well, here you see the consequences of hypertonic and hypotonic conditions. The upper right are red blood cells, and under hypertonic conditions, water flows out of the cell and the cells shrink. In isotonic conditions, the water concentration is the same inside and outside. The cells are happy. Under hypotonic conditions, water rushes into the cell, and if the conditions are severe enough, the cells will burst. On the right, the same thing is illustrated again, but comparing what happens in animal cells and in plant cells. On animal cells, they don't have a cell wall, and under hypertonic solutions, the water moves out of the cell, and the cell shrivels, whereas under hypotonic conditions, water rushes into the cell and the cell could burst. In the case of plants, they have a cell wall, and so if the cell shrinks, it's called plasmolysis, or it's turgid in the case of hypertonic. Tonicity in the plant cell vacuole with respect to the cell's environment. In a hypotonic condition, this is the solute concentration outside the cell is low. Water rushes into the cell and the vacuole swells, it's very large. And under hypertonic conditions, water rushes out of the cell, solute concentration is high outside, water rushes out, and the cell shrinks within the cell wall. Movement of small molecules. So we're going to compare how small molecules move across the membrane with how large objects move across the membrane. Well, there are three possible types of transport: diffusion, passive transport, and active transport. Those are illustrated here in this diagram. Diffusion is simply the movement across the membrane. Only certain substances can do that because they have to get across first the hydrophilic layer and then a hydrophobic layer, and then another hydrophilic layer. But some substances can do that without any help. A passive transport, the molecule gets help, but it doesn't require energy. It's facilitated simply by the presence of a protein in the membrane. Active transport on the right is transport against a concentration gradient. And that requires energy, usually in the form of ATP. So you see here, the blue balls are at low concentration inside the cell. They're at a higher concentration outside the cell. And yet the direction of movement is from inside to outside against the concentration gradient. And to do that, ATP needs to be hydrolyzed to release energy to drive these ions across the membrane from low to high concentration. So is this clear? Anybody have any question about the difference between diffusion, passive, and active? I'll elaborate on these as we go. So passive transport, no energy input is required. There are channels, proteins make channels in the membrane, and some are open and some are gated. They open or close in response to a stimulus. And all movement is always from a high concentration to low. Passive transport is always from high to low. So if you look at this drawing of a membrane, here's a channel, an open channel. If you look at the green balls, they're at a high concentration on the outside of the cell, and they move through the channel to a lower concentration inside. In the case of a gated channel, you look at the pale green balls, a gated channel will open on one side of the membrane in response to some stimulus, and then it will close and open on the other side of the membrane, again in response to a stimulus. But again, the concentration is going from high to low. The concentration of pale green balls is high on the outside, low on the inside. So the balls are moving from high to low through this gated channel. Active transport is entirely different. It requires that a cell expend energy in order to move molecules across a membrane. And the movement is always, I've underlined here, the movement is always against the solute's concentration gradient. Much of the transport in biological systems is active. And the energy, they always require energy, and that energy usually comes from ATP. But the functions of active transport, three main functions. Cells can take up molecules against a concentration gradient. Molecules so you have both uptake, and molecules can be moved out against a concentration gradient. And the cell can maintain the optimal concentration of ions by these opposing processes, uptake versus outgo. Cells can establish homeostasis. One of the most common active transport systems is called a sodium potassium pump. And there are some websites here if you want to see this illustrated in action, you can look them up, but we don't have time for them now. So what is the definition of the mechanism? Well, sodium ions are pumped out of the cell against a concentration gradient. And conversely, as that is happening, potassium ions are pumped in. The effect of this is that it lowers the potassium concentration inside the cell and increases the potassium concentration. Let me back up. It lowers the sodium concentration inside the cell. It raises the potassium concentration inside the cell with respect to the surrounding medium. And that's illustrated here. So the blue protein going through the membrane, that's the pump. So what you see is that there is a low concentration of the blue sodium ions in the cell, relatively low compared to outside, but the pump transports them against this concentration gradient from inside to outside. And ATP is needed for that to happen. If we look at the purple ovals, they're at a higher concentration inside than outside. But this protein, the very same protein, this is the pump, it takes potassium ions from outside and transports them inside, again at the expenditure of ATP. So if you eat too much salt and the concentration of sodium is too high in your cells, the sodium potassium pump pumps sodium out and then potassium in to compensate for that. Okay, one other point I want to make is that membranes, in addition to the concentrations of ions, membranes usually separate charges. One side of the membrane is usually more positive, and the opposite side of the membrane is usually more negative. So ions are transported across either with their concentration gradient or against it, and charges can vary, can be more positive or more negative, and this is called an electrochemical gradient, which is based on two things, the chemistry and the electricity. Chemistry being the ions, the electricity being the charges on opposite sides of the membrane. Oh, what you are familiar with already, the CFTR, the transmembrane regulator. It's a gated channel that requires ATP, and it actively transports chloride. So you see that illustrated here. Again, the CFTR is the green sequence. It makes a channel. The transmembrane portions make a channel across the membrane, and chloride ions are actively pumped out. And when you do your project, you'll become intimately familiar with this process. Okay. So far, we've been talking about small molecules, in particular ions. Now we're going to talk about large things, bulk transport. Two basic processes, endocytosis and exocytosis. We've already talked about endocytosis with respect to coronavirus. When it binds to a receptor on the surface of the membrane, the membrane invaginates and the pits form vesicles. Now the process can be reversed. If the cell wants to get rid of something, it can make a vesicle which moves to the membrane and fuses to the membrane and then is expelled. Exocytosis is the expulsion of substances. So for example, if there are breakdown products from salts or amino acids that the cell wants to get rid of, it can package them in a vesicle, move that vesicle to the membrane. The vesicle membrane fuses to the plasma membrane, and whatever garbage the cell is trying to get rid of goes out into the extracellular fluid. Now, endocytosis can be of three types: pinocytosis, receptor-mediated endocytosis, and phagocytosis. So pinocytosis is uptake of liquid. You can see that here at the top. The extracellular fluid is packaged and taken in a vesicle. Receptor-mediated endocytosis, where the substance binds to a receptor on the surface, that causes invagination, a coated pit, and a vesicle. So this is what happens with coronavirus. It binds to a receptor on the surface and enters the cell by receptor-mediated endocytosis. Well, phagocytosis occurs if the object is very large. So for example, supposing it's a bacterial cell. Well, the bacterial cell can bind or attach in some way to the plasma membrane, which again causes invagination, and ultimately the cell gets taken in a vesicle. So these three types of uptake of large things. And finally, I'm out of time, and this is the last slide. I'm not going to go through this, but it refers you to the textbook, pages 63 to 89, and is sort of a guide to the topics within this material that you should think about and know for upcoming quizzes and tests. So I will put the recording of this. First of all, the slide set is in Canvas. I'll send you the link to the recording. Any questions quickly before we finish? No? Okay, well,
Professor,
yeah.
for the quiz today, it's a redo of last week's quiz, right?
Excuse me, I have a question. I couldn't hear him for a workshop.
Wasan.
Slide.
There were two questions here. One was, there is no quiz today.
I don't know why, but I went on assignments and I don't know why, but it showed like last week's quiz to take again.
Oh, well, if that's the case, I'll have to check on that. If that's the case, that's a mistake.
Okay.
You're talking about quiz 5?
I could check right now.
Quiz 4.
Yeah, it's quiz 4.
Well, you have quiz 4 twice and you don't have quiz 5 at all?
Check. I guess it was a miss. Wait 1 second. Yeah, it just says upcoming assignment quiz 4 mutation transcription translation. And then I already had my score, but then it says take the quiz again.
Okay, I gotta look into this. I'm looking at the grades right now. So almost everyone has taken quiz 5 and quiz 4. So has anyone else had a problem with seeing quiz 4 twice? Well,
No.
yes.
No.
Okay, well, I graded all the quizzes that have been turned in.
Yeah, I already
Quiz.
I already have a grade for it.
Okay, well, I'll check and see why it's showing up for you twice. It shouldn't be. And if there's something wrong, I'll correct it.
Okay.
That, and it's quiz 4 that you're seeing twice, right?
Yeah.
Okay.
Okay, well, there's no more quizzes for Monday right now?
No, there are no more quizzes scheduled for now.
Oh, cool. Okay. And we already, the homework that we had was turned in for today, right?
Yeah. Yeah,
Okay,
and
so that's it.
I'll go over that next time.
Okay.
The answers to the pancreas.
Okay.
Okay,
That
and
it?
you have another worksheet on membranes, and you'll need to know about tonicities, osmosis and diffusion in order to do the problems. Okay, that's it for today. Thank you all. And unless there are more
Have a good
questions, I
weekend.
will end
Thank you,
the,
Professor.
Have a good weekend. Bye-bye, guys.
okay,
Thank
bye.
you.
Thank you, Professor.
Bye.
All right, see you next Monday.
Professor, I'd like to talk to you for
See
a little
ya.
bit.
Okay, I'll leave the,
Bye.
bye. I'll leave Zoom on if anyone wants to talk more.
Okay,